So given all those pieces, I want to put them together with some particular for instances. And so I want to start with bacterial nutrition. And this is very variable. And I'm going to give one example. So people think they have different diets if they're a vegan or a vegetarian or maybe the paleo diet or whatever. That's a tiny subset of possible diets. There are bacteria out there that get their nutrition from mercury or from plutonium or from arsenic or from whatever kind of cruddy things they can find in the mud. Bacteria, it turns out, have a huge range of metabolic diversity. Some of them live on hydrogen sulfide and oxygen, the kinds of things that would be toxic to us. And so I'm going to talk about one kind of bacterium, which has, as bacteria go, relatively boring nutrition. But if you take a microbiology course, I'll teach you about a lot more interesting and cool ones. But it's just an idea of how things are simple. So this is the intestinal bacterium called E. coli, which is was famous in the past as a research organism. You guys used it in Bio 111. It's also famous as there are strains of it which are quite nasty and pathogenic. It lives, all of us have lots of these living in our intestines. It's sort of a normal part of our intestinal, what they call microflora. So how does it make a living? So here, if you draw the cell, there's some basic anatomy of these guys. There's a plasma membrane. There's DNA is free in the cytoplasm. These guys have no nucleus. And they've got around them a cell wall. And that's protein and carbohydrate. Okay. And every other living thing, they've got growth and living. They need to do that. Just like every other thing, they've got to get all those pieces. And so they take in oxygen, and they give off CO2, just like every other thing that does respiration. They need monomers, they need glucose, and they need vitamins. Right? So that little bit there is in common to every living thing. How do bacteria feed that core? Their food is glucose, sulfate, phosphate, and nitrate. So these guys come in. You can grow them on many things, but the minimal thing is biosynthesis, and this is many reactions. So those things come in and come out as monomers, glucose, and vitamins. And if you remember, for those of you who are in my flavor of Bio 111, there was that pathways chart. The thing looked like a big subway map. I don't know if you remember from way back at the beginning of Bio 111. That's actually a small subset of, when I say many reactions, there's thousands. That they take simple stuff like this, shuffle it, rearrange it, connect it, reconnect it, move it all, change it around, and out comes monomers, glucose, and vitamins, which is what they need to live. So you can feed them very simple stuff. The bacteria are very capable biochemically. Basically, they make all it needs from these. There are no polymers needed. Turns out that big subway map of reactions that bacteria can do, many of them we can't. That is, we are biochemically much less capable than bacteria. We'll come to why that is in a second. But bacteria can make all they need from those very simple ingredients. When you grew them in lab, we gave them a break and provided them with a variety of monomers and things, but you could actually grow them on very simple stuff. Questions about that before we talk about some other examples?